And my title is something unique. It is particularly focusing on a particular topic. It is not a general topic. I am going to brief before you the lead we have with respect to the Ideotype. So medicinal plants are altogether different than say food grain crops or say other crops, because in medicinal plants what we need is the concentration of active ingredients. For example, for rice we will be talking about more grain yield. In wheat we are also interested to harvest more grain yield. But when it comes to medicinal plants, we will be always focusing on higher biomass or say higher active ingredient. So both together makes active ingredient yield. So for the crop improvement perspective, a person who is working in medicinal plants, he will always be focusing on say what is active ingredient yield in which particular genotype or which particular accession or the proposed variety, the active ingredient yield is higher in which one, and that will be proposed further. We are having 26 AICRP centers across India, and if any of the promising material we find, we propose it to conduct multi-location trials through across our 26 AICRP centers for at least 3 years. And based on those data only, we propose any variety for that particular region or through across the India, based on the data of 3 years across the India. We are also, I would like to inform you that we are also a dust testing center, and we have developed, our directorate has developed dust descriptors of this Andrographis paniculata, which has been adopted and has been notified by PPV and FRA, and we are authorized dust testing center at our directorate in Anand, Gujarat. So what is an ideotype? What I have already told you, it is altogether different than food grain crops. Then what is the significance of flowering in this particular crop? Before that, we will see why this plant is important medicinally. So it is an annual herb, and it is also known as king of bitters. People used to say nothing is bitter than this particular crop. That's why it is being called as king of bitter. It is very kadwa, and it is distributed across southern Indian, India and Southeastern Asian countries, including India, Bangladesh, Sri Lanka, Hong Kong, Indonesia, Myanmar. So this is geographical distribution of this particular Andrographis paniculata. It is used for say alopecia, boils, chronic and seasonal fevers and cough, diabetes, dysentery, dyspepsia, so gonorrhea. It has many roles to play in different kinds of diseases and disorders, including irregular bowel habits, jaundice, leprosy, skin disease, scabies, and even in snake bites. What Professor Rao today, just before this lecture, said that ethnobotanically, so people used to consume the entire herbage of this particular plant, okay, for the through decoction. But say on a chemistry side, it is containing terpenoids, say more than 78 ethnolabdane diterpenoids. It contains more than 40 flavonoids, rare noriroids, steroids, and other compounds are there in this particular medicinal crop. Cultivation. Farmers used to cultivate this crop. It is raised through transplanting. Average 45 days of seedlings are being transplanted, and the crop is raised for 150 to 160 days. After transplanting, it is being harvested. The active ingredients, say endographloids and neo-endographloids, they are at peak at 120 days after transplanting. But what happens? At this particular stage, the farmers do not get the herbage yield. So the cultivation is not a profitable business for a farmer if he harvests it at 120 days. So he will have to wait for 150 and 160 days. And if it is being harvested at 150 and 160 days, a farmer can get around 3500 kg of dry biomass, aerial dry biomass. Now look at the fractionation of, say, functional allocation or say biopartitioning of this endographloid. Leaves contains 3.5% endographloid, whereas stem on an average contains 0.8% endographloid content. And the entire herbage together at the time of harvest, nearly about 1.5% of endographloid is there. What is the impact of flowering on economic cultivation? Now see what happens when the plant transits from vegetative to reproductive phase. The aerial part or say the tip of the plant is transformed into inflorescence. So the plant is an indeterminate type. It becomes an indeterminate type. And the aerial biomass, what we see is actually bushy, not boozy. There is very much air spaces, just the aerial tip is elongated, and that's why the plant seems to be a bigger plant. Otherwise, the biomass is not in proportion to what we are seeing. The plant seems to be boozy, but it is not giving that much total herbage yield. So at maturity leaf, the another issue is that at maturity time, the older leaves shed, they drop down, and new leaves do not come. So what in the previous slide we see, leaves contain more than 3.5% endographloid. Stem contain only around 0.8%. So if at the time of harvest, we are harvesting the material which has lower leaf content, then but natural our total product will have low endographloid yield. So an ideotype in this particular crop will be such a crop which will have higher number of leaves or higher leaf biomass at the time of harvesting. But the actual problem is that once the flowering starts, newer leaf stops. So to overcome this particular problem, we need to have such a crop in which, such a plant in which the leaves continues to grow and the older leaves do not drop down. So this is a kind of an ideotype. This is a released variety of Vallabh Kalmeg 1, VK1, released by our own directorate, and this is an ideotype. So plant with a higher leaf content or a stay green type of plant, a plant with higher endographloid content at harvest maturity, say for example at 150 days to 160 days after transplanting. So this kind of plant will give you on an average higher and stay green type of material at harvest time. Unfortunately, what happens, farmers used to harvest it at 160 days time, but at that time leaves, contribution from leaves comes as a lesser content. So flowering requirements in this particular crop. I have been working in this particular crop for since 2018, and my own experiment says that this particular crop requires at least 58 days after transplanting, including 40, in addition to 45 days of nursery stage. So if you give at least 58 days after transplanting, the plant will initiate flowering. The another requirement is that growing degree days. Growing degree days requirement varies from 828 to 2428 degree days. We are having 400 accessions available at our own directorate, and this is an average data of our directorate. It is a short day plant, and it requires, it is raised in Kharif and flowers in winters, and it requires critical long dark periods. Say for example, more than 12.3 hours of dark period initiates flowering in this particular crop. So if your crop is raised in July or end of the July, it will initiate in September, around September time. As I said, dust descriptors. This particular crop has dust descriptors, and this is, see, the rachis types are dense rachis, or else it may have loose rachis, and the inflorescence is panicle type as per dust descriptors of this particular crop. But what we have identified in 2018 one plant was there in 2018 which was not flowering at all. So we maintained that plant for up to 2013. I'm a physiologist. We tried many physiological interventions to initiate flowering. And 2022, sorry, not in 2023, in 2022, we got success. And we were successful in initiating flowering through one of the physiological mechanisms. And that physiological mechanism has initiated flowering. We got nearly 2000 seeds, and those seeds were transplanted, nursery was prepared, and those seeds were transplanted in the field. But this particular type, we can see that this is not following the dust descriptors. And Ideotype, what happens? When the inflorescence is started, it actually starts from the apical tip. But in this particular crop, this behaves like an indeterminate plant. Flower comes, but on the auxiliary bud, not on the apical tip. So this results in a kind of stay green plant. This continues to produce newer leaves till 150 days, 160 days, or even 180 days. We may see that after flowering, the plant type has been changed. Nursery was prepared, and these plant seedlings were raised in the field conditions, on field screenings. Then what happened? We were trying to test that whether our physiological intervention was really working across these 2000 plants or not. So we tested different mechanisms, or we tested different. We employed this different mechanism to initiate flowering in this population also. We have grown these seedlings in actinic light, and then we have transferred to the field. We have tested different chemicals. Then we tried a mechanical induction of flowering also through transferring the concept of florism transfer, and we got success in this population also. This was the first flower bud, and then the flower. What I said, that on the apical tip there won't be any flower in this particular case, say non-flowering type of plant. The closer view. So this was the story, what we found, but our ultimate intention is to find out why this type of flowering habit is there in this particular plant. So we performed, but we found that there may be 2 to 3 basic reasons related to genetics, and there may be an environmental factor, say lack of appropriate photoperiod, temperature, or stress conditions. But it was not working because other plants, other 400 accessions was raised in the field conditions and they were flowering, but this particular plant, or the progeny of single plant progeny of this particular plant, was not flowering. So that was overall environmental factors. Then it may be a genetic variation, or genetic variation may be because of the silencing of FT gene or the regulatory genes involved in these particular flowering mechanisms. Then maybe epigenetic modifications also, and because of the regulatory network of say CO and FT gene expression. So to identify what the actual issue was at the molecular level, we performed a transcriptome, comparative transcriptome. And this case, in 3 biological replications, we have generated more than 111 GB data. This 111 GB data, raw data, was then transformed to, or then cleaned, and high quality data was used for say transcript predictions, and then unigene prediction, and then CDS predictions. This was the flowchart how we have performed our transcriptome analysis, say for comparative transcriptome analysis. And we have also identified, we have tried to identify some of the microRNAs, because microRNA 156, 157 and 172 has a great role to play in controlling flowering. This we can see that the combo assembly has the highest length, 100,000 to 5,000 base pairs, and this is the data of the assembly. And then we have predicted CDS, coding DNA sequences we have predicted, and based on this 100 and all the 9 samples, we have performed a master assembly, and this master assembly gave us 2.68 lakhs total CDS, and out of this 2.6 CDS, 2.17 CDS got hit against NR data. And this is the distribution pattern of the CDS, and we may see that 200 to 300 and 300 to 400 were the highest number of CDS available in our combo assembly data. The highest number of genes were against general function predictions only, and then signal transduction mechanisms. And all 4 protein databases were validated to confirm our protein data, and we can see that the 54,266 genes were validated through NR, Uniport, PFAM, and COG analysis. And then it was the transcriptome was compared between flowering and non-flowering plants, and we found that there were total 9.8 thousand CDS which were differentially expressed, and significant differential expression was found in 15.1 thousand CDS, out of which 7.6 were down-regulated and 7.4 were up-regulated. This is top 50 genes expression profiling. And based on this transcriptome data, we have prepared 5 putative pathways of say flowering control in Andrographis paniculata, and all these CDS we have found, and these CDS have been deposited to NCBI database. Because the central pathway or the central molecule or the central gene is FT, we have focused more on FT gene. This is the protoparam amino acid composition of FT protein. A model, in silico model, has been developed for this FT protein through iTaser software. And then phylogeny tree was predicted, and the predicted protein was very much similar to the already available Andrographis paniculata FT gene, and it was very closely related to Salvia hispanica. So now we were attempting to find out say microRNAs which may have the role in masking the FT gene, so that the flowering was disturbed. At read level we tried, but we did not get any success. But at Unigene level we found 10,414 sequences representing these microRNAs, and we kept parameters that the word count should be 17, not less than 17, and the E value should be 1000, and maximum target sequence should be 100. And stringent criteria were also kept to end-to-end alignment with query length and 100% identity, 0 mismatch, 0 gap. So based on these criteria and parameters, the data were filtered, and we reported 1926 microRNAs from this 111 GB transcriptome data. And the precursor molecules, precursor structures and hairpin structures of the microRNA 156 and 172 were also predicted, and their free energy level was argumenting, what I can say that based on this free energy, we considered that these were the possible microRNAs. Their hairpin structure was also validated. These are the 2 microRNAs, 156 and 172, and as I said that 156 and 172 have already been reported their role in controlling flowering genes. So microRNA 156 expression is higher in leaves during the juvenile phase and declines after the phase shift, whereas the expression of 172 follows opposite trends in our particular data. And we found that the microRNA 172 was targeting the epitela gene API2, whereas 156 were targeting other genes, 7 genes. And epitela 2 is the gene which actually defines the site of meristem transformation to flowering. So this was the reason why the plant was unable to identify the apical tip to be the site of flower initiation. This is our work, and now we are working on validation of this particular aspect. So from this study, non-flowering and a stay green type Ideotype with higher Andrographis yield we have identified, and we have successfully induced flowering in a non-flowering type plants. A huge transcriptome data was generated, and we have deciphered 5 putative pathways of flowering. We have reported 109 genes involvement in controlling flowering, out of which FT and Sapitella are the key players, and MI172 as well as MI156 is also playing a major role. 156 is playing a major role in masking FT gene, and which primarily seems to be the reason behind this non-flowering type behavior in this particular Ideotype. Thank you.